So in this drawing, we sort of pick up where we left off with the zygote, and we want to see what happens to the zygote. Now, I've made a note right here at the top for you. It says the zygote is a totipotent cell. Toti means totally, and potent means powerful or capable. So in what sense is this cell totally powerful? Well, it can turn into any type of human cell, even supporting cells such as the placenta and the amniotic sac. All the DNA in this cell, this first zygote, is open and accessible. None of it is methylated or closed in any way. As the embryo develops, the cells will become less and less potent. As we go along, they'll have much of their DNA closed. So at this point, this cell can be anything. And you need to remember that in embryology, some of the cells that come from the zygote won't end up being part of the baby. They'll end up being part of the placenta or the amniotic sac or the chorionic sac or the yolk sac or the allantoic sac or something like that. So we're going to draw day 1 again. Let's zoom in on day 1 and day 2. And day 1 is simply the zygote, and we saw in the last drawing how the male and female gamete, the sperm and the egg, came together to make a complete set of DNA and make the zygote. So in day 1, we're just going to kind of shade this inner circle. Now, this outer ring, in fact, we can just go ahead and label it, is going to be our zona pellucida. So you'll remember that from the drawing we did on fertilization. Well, the zona is still with us. Now you can see all that corona radiata, all that fancy fiery looking cells around the outside. So that's kind of gone now. And we're just left with the zona pellucida. And remember, lucid means clear. This is kind of a clear layer. And you can kind of think of this as the shell. You know, if this is an egg, this is sort of like the shell. And strangely enough, we'll see by the end of this drawing, just like any egg has to hatch, a human embryo has to hatch out of its shell. Kind of funny, but true. So let's give it a nucleus. Lightly here, and you can write nucleus if you're afraid you're going to forget, or you can leave it unlabeled. And let's make sure we know this is the zygote. Remember, zygo means joined, so that's what you get when you join a male and female cell. You get a zygote. Okay, and we just made it look a little bit 3D here to remind us that it's not flat, but it is in fact a sphere. So what happens between day 1 and day 2? Well, it takes an entire day for this cell just simply to divide in half. So if you want to make a line right about here, and I'm going to round this a little bit, say kind of, so it looks like there's 2 things sitting in here. And again, I think I'll kind of shade this a little bit so it looks a little round. On this one, you can shade the top or kind of the underside, or a little bit of both. And if you don't want to shade, it's okay, it'll work all the same. You want to add a nucleus in each. Okay, so now we have 2 cells. And as far as I know, there's no fancy name for this, just the 2-cell stage. However, there is a name for what happens here. When a cell splits in half but stays together, okay, we call this, right down here on this line, we want to write the word cleavage. And I'm going to write it in caps because it's so very important. C-L-E-A-V-A-G-E. To cleave something is to split. So cleavage means splitting in half. Now, this is different from binary fission, you know, when a bacteria divides in half, or when a paramecium splits and makes two of itself, it becomes two separate individuals. So these aren't just two cells sitting together, all smashed together. There are actually little stitches keeping them together. It's like they're sewn together, right? So if this popped off, these things wouldn't come apart. They're stuck together. And we're going to take an up-close look at one of these over in this space here. So let's circle one of these and go like that to show we're going to take a look at it. So we need some special junctions. A place where things come together is called a junction. And there's different kinds of junctions between cells, and we'll see about 3 or 4 different kinds here in the course of our whole course. And this one we want to meet first is called a gap junction. Now, these cells need to stay in communication with each other because there's going to be some decisions being made. The cells are going to decide, now you do this and I'll do this. Some of them are going to become one thing and some are going to become another. And cells need to communicate. And one of the ways they're going to do this right now is through these gap junctions. Now what I've drawn here is 2 plasma membranes sitting side by side. So you notice the little heads and tails here. So this is going to be part of a cell wall here. And this is going to be part of a cell wall. Now I've left a gap because we're going to draw a little gizmo in here. This is going to be a little kind of protein gadget. And so we want to draw a line here and here. And then like this. Now of course this is much simplified, right? So as you're learning as we go, when we draw protein gadgets, we tend to oversimplify them. We make them into circles and squares and triangles. In reality, they're a lot more complicated. Now, we're going to be looking at the side view in this. This is a side view. And then we're going to make a little dashed line in here to indicate that what we can't see is that there's like a tube, a hollow place running through the middle. So if we were to draw an arrow, I can make the arrow dashed like this. This area here is like a tube. It's going to be kind of like a little sewing bobbin. If you know anybody that sews, or if you sew and you use bobbins in the sewing machine, that's kind of what this is going to look like. You're looking at a kind of a side view. In fact, to be accurate, we could probably kind of go like that. All right, so this will become clear in this next drawing. Right here, you need to draw an oval like this. This is going to be a fun little drawing. And then underneath, draw another oval. Just sort of imagine that they're almost overlapping there. Are there this. And then draw a circle here, a little oval. And then you want to come down like this, straight down, but don't draw on this one. Just hold your pencil above the paper until you get to this line and then draw like that. And then the same thing over here, like that. And then you want to draw the same round shape right here. You want to draw that right here like this. Alright, so I want to be able to see that one there. Okay, and then shade this a little bit like this. Alright, so you can already see, ooh, it looks like it's a tunnel, doesn't it? And then we want to shade this a little on this side and this a little here. Okay, and let's, just for fun, put a little shadow like this coming out from here. Okay, so that really looks like one of those bobbin things, doesn't it? And then we can kind of a little bit shade around here, make these look a little bit thicker maybe. So what you're seeing to the left now, does it make sense that you're looking kind of straight on to this thing? So if you're looking, you were looking like this. So this would be like this thing, right? And this thing here was in the middle. And then this bottom one would be like right here. And then that's the tube. We can even make our arrow like this. And then on the other side, like that. So it looks like the arrow's going through. And then let's make a note for ourselves that cytosol, the cell gel, the gooey stuff, can go back and forth. Can go back and forth between the cells for communication. Now, in the future, the cells are going to have a lot fancier methods to communicate. They're going to have more options, but right now, this is their primary option. This cell could make a chemical that says, I'm going to become a placental cell, and it would leak over here, and this cell could either be programmed to say, oh, well, I'll do that too, or, okay, I'll go do something else. So you've got messages leaking back and forth. Now we're going to see gap junctions again when we get to heart cells. Cardiac cells, or the cells of the heart, are special in that they really have to do things together in unison. If these are cardiac cells, they both have to function at the same time. And so we have all these little gap junctions between cardiac cells, and this is what enables the heart cells to beat together in unison. They all function together, and it's gap junctions that allow them to do that. Now there will also be some tight junctions, which are more like snaps. So if we're using sewing analogies, and these are little bobbins, there could also be little snaps in between here. They're called tight junctions, and they just snap things together. There will be some tight junctions as well as these, but we're going to kind of skip over that. We're going to come back to different junctions when we get to other types of cells and tissues. We're going to come back to junctions again. But I just wanted to mention that right now because that's how these cells are communicating with each other. So let's go down here and see what happens next. So somewhere in day 2, probably, and there's some variation. Some people might say day 2, day 3. It doesn't really matter. About day 2.5, let's say. This thing splits again and becomes 4. So that top one is going to split, and the bottom one is going to split. Once again, remember this outer ring is the zona pellucida, and that's going to be with us through this whole first week. That thing stays right there. Now the zona pellucida is not really flexible, so you notice now that we have 4 cells, these things are getting smaller. Now in your drawing you probably have this note down here. It says cells are getting smaller while overall size is staying the same. So I already jotted that for you. So I'm just going to put 4 cells. All right, so day 3. Now each one of these cells is going to divide again. So we have 4 times 2 is 8. Now at this point, it's going to get a little more complicated to draw. Let me show you a picture first. Some actual photographs of this process. So here we have the zygote. This is also just the zygote. And here's where we go from 1 cell to 2 cells. There's the zona pellucida around it and the 2 cells. And then here we have 4 cells. Now you know this is an actual microscopic view, so we can't choose how the cell is turned. It just so happens the way that it's turned in this picture. Here's one, and these two are kind of behind it. So the cells are clear, so we're kind of seeing through these two. These are on top, and these two are kind of on the bottom. So cells aren't always turned the way you want when you see them. And then here is, let's see, 1, 2, 3, 4, 5, 6, 7, and then this fuzzy one is 8 behind it. So again, we are looking through a layer of cells, and I'm not sure whether this one is on top or this one. And the thing about looking under a microscope at something 3D is that you can only focus on one layer. Like if I wanted to focus on this roll of masking tape, I'd have my choice of focusing here, or here, or here, or here, or here, or here, or here. As I rolled the microscope up and down, I wouldn't be able to see the whole roll because it was too large. So that's what's going on here. We're seeing a little bit out of focus, and then this layer was actually exactly in focus, and then the things on the bottom again would be slightly out of focus. But there is the 8 stage, and then soon we're going to see it's either getting smaller again. This has maybe 16 or 32, and then we're going to move to something very different where it starts to have a ring. I'll get to that in just a minute. So let's draw the 8. So make them about this big, 1, 2. Yours doesn't have to look like mine. Just kind of do your best to draw 3, 4, 5, 6. I'm going to make 7, 8, kind of peeking out a little bit there. Okay, and we'll give them some nuclei. I'm going to shade a little bit around them. I guess they really probably would be like hitting the edge a little bit, but eventually we are going to have some kind of empty space opening up. Okay. There we go. Now we'll write 8 cells. And the note here, this is a critical stage for unknown reasons. Some embryos don't make it past this stage. And the reason that we know this is because of in vitro fertilization. When a woman has difficulty getting pregnant, sometimes they'll take egg and sperm and they'll mix them in a test tube and try to have fertilization occur in the test tube. And then meanwhile, this is happening. This will happen outside the body in a test tube. Kind of weird. And then, so you'll have a bunch of these in a test tube, basically. And then they will freeze them at this stage. And then they can get them out of the freezer, thaw them out, hopefully they'll continue growing. This surprisingly actually basically works. But because they do this so much, scientists have looked at this little 8 stage, probably some 4s and 16s also, but they've looked at this 8 stage so much that they actually have a lot of data, and they know out of every 1,000 or every 100 of these, they know how many will make it and how many won't make it. Sometimes, even freezing aside, forget that, sometimes they've noticed that these things just don't ever grow beyond this 8 cell stage, and they don't know why. So this is where life ends for some embryos. Most of them go on, but some can't make it past this stage, and maybe they'll figure it out someday, but right now it's a mystery as far as I know. Okay, just a little bit of embryo trivia there. Okay, day 4, what's going to happen here? Well, do the math. If these all divide, we're going to get 8 times 2 is 16. So we're going to make kind of a nondescript thing, and then you can guess after 16 comes 32, right? So let's just put kind of like 16 to 32 cells, and you're just going to make a clump. And we're not going to worry about counting exactly. We'll have a few peeking out behind the edges here. Okay. Now this stage actually has an official name. Except for zygote, none of our stages up till now have really had an actual name. You can put some nuclei in there if you want. This thing is called a Morula, and it means mulberry. Can you believe that? You know, like a raspberry. Got a picture of a mulberry here. There's a mulberry. So you can kind of see the resemblance, right? Put a little stem on it there, and got a little raspberry or mulberry. So the special note I want to make about this, besides the fact that it has an actual name, is that now we're getting enough cells here. If you imagine, this is a solid ball of cells. Well, oxygen is going in like this. Carbon dioxide is coming out. It's using diffusion. Remember we covered that a long time ago. Diffusion is when the gases just go right in and out. So these outside cells that are near the wall, no problem. They're getting lots of oxygen, getting rid of their carbon dioxide. But what about the cells that are right smack dab in the center? They might be, 3 cells away. The oxygen would have to go all the way in here. And these cells on the outside would grab it first. So cells that are more than 3 or 4 or 5 body lengths, cell lengths, away from the oxygen source are going to start struggling. And in our bodies, our cells are never more than 1 or 2 cell lengths away from a capillary. So we have this extensive little capillary system that delivers oxygen to each and every cell. Well, here we don't have any capillaries yet. The only thing we have is diffusion. So we've got a problem. How are the cells on the very inside going to get this oxygen? No capillaries yet. Let's just write a note right here. Inner cells will start to have trouble getting O2. Now, as far as nutrition, the cells are still running on all the energy that the egg had saved up. Remember, the egg cell had all this stored up fats and sugars and proteins, and that's getting divided up, but these things still probably have that original energy inside of them. That's going to run out pretty soon, but right now they're still okay for food, but it's oxygen they're struggling for. The solution is to make the ball hollow. So we want to draw day 5. It's starting to go like this. Now this is kind of going to be more of a cutaway. On the exterior, it wouldn't really look any different than this. It would just, the cells would look smaller. So here, let's make it look like there's some going into the distance like that. And then let's do something tricky with shading. Go in here and kind of very gently shade a little bit like this. Alright, does that make it look a little bit hollow? You can kind of see it's a cutaway. Can darken a little bit around here too. There, it looks sort of hollow. And if you want to remember, this is a cutaway view now. So this is maybe 32 to 64 cells or so. And this has a name also. This is called the blastula, or you will also see blastocyst. So So either word is fine. Don't let it throw you. Blastula, blastocyst. Don't worry about it. Now you'll see this term blast a lot in embryology. We have the blastomeres and the blastocele and all kinds of things that have blast in it. Now, of course, this word blast has nothing to do with dynamite or explosions. It actually means bud, a shoot or a bud in a plant. So let's make a note here, another parentheses. Blast means, quote, bud. And of course, the main thing to know about a bud is that it turns into something else. You have a flower bud and a leaf bud. It opens up and it turns into something else. So the bud is kind of like the little baby part that then develops. Now, if your drawing is good and it's obvious that there's like an empty area in there, you don't have to add this note, but if you're worried about remembering, you can write, there is a cavity, that's what they call it, an empty space like a cave, a cavity filled with fluid. And the fluid would be mostly water with some nutrients, obviously oxygen and things. But if you want to add that note. But if your drawing is good and you can remember, you don't have to add that. Okay, let's go down and see what happens on day 6. We're getting towards the end of the first week. And the first thing you'll note is that our zona pellucida is still with us. This good old ZP, it's hanging in there all this time. And we're still going to have that ring of cells that we drew for day 5. So this is still called the blastula, or the blastocyst. Pretty soon you won't have to draw all these cells anymore. I'll draw them for you, but right now it's good to draw them so you remember they're there. And we're going to add something though. On day 6, we have some cells that grow like this. A little lump of cells grows like this. And I'm going to kind of outline mine like this. You can kind of see that there's like an inner area there. And this has a very easy name, fortunately, for once. So don't make it too hard. This lump is just called the inner cell mass. It's just a mass or a lump of cells. And they aren't any particular kind of cell yet. These cells, and when I say in the blank down here, these cells, I mean these, if you want to point to them. These cells are, and the word you need to fill in the blank here is pluripotent. P-L-U-R-I, pluri means many, and then potent, we already met, means powerful or capable. Now, these are not totipotent, not totally capable, but these are pluripotent, many capable. So this is just a tiny, tiny step down from totipotent. But they're still very flexible cells that can turn into many different things. So therefore we call them pluripotent. And you also see in the note that these are cells that are often harvested for embryonic stem cell research. Now, the use of these cells from human embryos is very controversial, as you probably know, because this is capable of becoming a person. I mean, you looked like this at one time. Now, the nice thing is that there are lots of different animals, as simple as the lowly sea urchin in the ocean, all the way up to mice, who have this same thing going on. Embryology is very similar in mammals, and of course we're a mammal, so you can use mouse embryos and do research all day long on their pluripotent cells, and nobody screams about it. And you can even use some invertebrates, and you can learn a lot. In fact, most of what we know about embryology has come from studying things that aren't human, because the processes and these stages are just so similar. Okay, let's go over to day 6 or 7. And I've called it 6 or 7 because when you get to this stage especially, you'll notice if you read a little bit on the internet and you go to a couple different sources, someone will say it's day 6, another place will say it's day 7, someone will say it's day 8. A lot of times they'll say day 6 to 8. You know, it can be different for each individual embryo, right? We have a general guideline, but for some embryos it's on day 6 and some it's on day 8, maybe even in a really unusual case it'll be on day 5 or day 9. But on average it's about day 6 or 7. And what happens here is that it finally hatches. Like we've been waiting for this zona pellucida. It's just got to go because these cells keep getting smaller and smaller. You're going to end up with a microscopic human being if you don't get rid of the zona pellucida. We got to get out of here if we're going to develop. So this is what happens. All right, so let's make a letter C sort of kind of right here, real lightly. This will be our guideline. Okay, we can go about over here, kind of make a very, very light C and kind of come up like this a little bit, like a fancy C like that. Okay, and then in here, we're going to make sort of a pear shape. So it's going to be smaller. In fact, you know what, let's just do this first. Let's go ahead and trace around here and make this look like it's thick, like that. And then in here, smaller, and then as it bubbles out, getting larger. Alright, so this is going to be the blastula popping out. That kind of looks like it's popping out. Okay, so this is our good old ZP again. And this will be our blastula, blastocyst. And we'll put our inner cell mass right here. Okay, so this is our, you can call it blastula or the blastocyst. I guess blastula is sort of the stage. It's the blastula stage, although sometimes they do point to it and say that's a blastula. More often the blastocyst will be what they actually call the thing, but either one is fine. So you'll see the note down here that the blastocyst secretes enzymes that soften the zona pellucida. So that was programmed in the DNA that at this point in development, on day 6 or 7, all of a sudden the cells start to secrete an enzyme that softens this. And then suddenly it grows. There's a little explosion of growth. And all of these will go through mitosis together, and boom, the blastocyst will get larger very suddenly, and it happens suddenly enough that it pops the zona pellucida open. And right here it looks like it's popped open, and then this thing just squeezes out and hatches. So we can put here the embryo hatches. So you hatched from an egg a long time ago, just like a chicken, right? Now, just for fun, I've listed some amazing facts for you down here. The first week is the same for all placental mammals. That means any mammal that doesn't lay eggs, so the platypus is out, okay? But for most mammals, this first week is the same regardless of how long the gestation period is. Now here's a good word to know if you don't know it already. Gestation, that means the amount of time it takes to produce a baby. Technically, it also includes a little bit of time before fertilization. Like for humans, there's 2 weeks between ovulation and fertilization, and those 2 weeks are included in the 40-week gestation period. But basically, we use the word gestation to mean the time of pregnancy. So for mice, the gestation period is 3 weeks. For elephants, the gestation period is 2 years. The first week is the same for both. So just think about that. If you have 3 weeks to make a baby, and in your first week you went from 1 cell to 2 to 4 to 8 to 32, you had the morula, the blastula, and you got to this where you have a little blastula hatching out. And in 2 weeks you have to go from this to a baby mouse. That's pretty amazing. For elephants, no problem. You got a whole 2 years, no problem. But for the small mammals, now, of course, when a mouse is born, it doesn't have hair and its eyes aren't open, so it's kind of half formed, so that's maybe a little bit cheating. However, there's some other mammals with some pretty short gestation periods and they come out completely formed. So it's pretty amazing that this first week would be the same. You think that it would be a lot shorter. Second amazing fact is that some mammals can pause pregnancy at this point right here and hold the embryo for several months, waiting until the right season. And examples of this, if you want to jot them down, would be bears, kangaroos, and the roe deer, R-O-E. A roe deer can do that, and maybe some others, but those are the examples that I read about. Now, before we go over here and fill in this map of where this is happening, I just want to show you a couple more photographs. This one here, this is obviously getting to the blastula stage, where it's got these cells around here and a cavity is opening up. I'm not sure what these are pointing to or here. This, I think, is pointing to the outer ring of cells, and I think maybe this one is pointing to an inner cell mass starting to grow. This looks to me like the hatching picture, doesn't it? Kind of where it's kind of popping out, and maybe this here is the zona pellucida, and this is the end that's coming out. It doesn't say that, but it's what it looks like to me. And then this would be the blastocyst out of the zona pellucida, and you can see the individual cells here, and that would be the inner cell mass right there. And I have a much better picture of this right here. That is what we just drew. Inner cell mass, and it looks like the zona pellucida is still around it, and then these inner ring of cells. And you can kind of see on that one drawing we tried to draw a little bit showing you're looking into the ball, and these are kind of the inner ones, but this is the cutaway view right there. So there's a real one. Now let's take kind of an overview and find out where this is happening. And I'm sure you know generally where it's happening, but we're going to find out exactly where it's happening. Now I want to start with a picture. You're probably familiar with this already. You know generally where this is happening. This is inside the female reproductive system. So this is where it's located in the body. Here we have a close-up, and of course you probably already know that you have the uterus, and these are the fallopian tubes here, and these are the ovaries. And what we're going to draw is just a section. We're going to draw one of these, and then kind of like this. We're going to kind of draw, and then you'll just see the top section of the uterus, and we'll find out where these stages are occurring all along here. So the problem is going to be that we have kind of a square space in a drawing that kind of wants to go out horizontally. So we'll do the best we can. Let's draw sort of a J shape like this. And of course, this does not have to be exactly like mine. There's a lot of different pictures online that I looked at, and each picture had a different shape for what we're drawing here, so the exact shape isn't super important. So let's then take this kind of up like that, up and down, and over like that. Okay, and then make this into a tube, and at the end we want to put what looks like fingers. kind of a raggy end to it. It's like a hand with a glove on the end. Come down here and just kind of come down like that. Okay, and then here let's make this, let's do like that. Something like that. And I want to add a little bit of color. This is going to be the lining of the uterus, which is called the endometrium, and it's very rich in capillaries and little blood vessels, which is really good for a developing embryo. In the next picture, we're going to see a close-up of this, so we might as well kind of draw a little bit of attention to it right now. And this is the part that builds up every month, and then it's shed and it builds up again, at least in humans. Now, remembering that this whole science of embryology is very similar in all mammals, but the timing is a bit different in the different kind of mammals. All right, let's put a circle right here. And then let's add a little, so this isn't just floating, this is actually tied. There's a little ligament that ties this, and this is the ovary. We could go ahead and label these things ovary, and we could put some eggs in here. Just some little circles, that's good enough for right now. And we can go ahead and label this. This is the FALL, F-A-L-L-O-P-I-A-N, fallopian tube. And I've made it look nice and fat here. In reality, it's pretty skinny. Diagrams are usually out of scale and out of proportion, but they help us to understand what's going on. And then we'll label this down here is the uterus. And if you want to label the red lining, is actually called the endo, E-N-D-O, metrium, M-E-T-R-I-U-M. It's a very Latin looking word. And I just put it in lowercase here. I don't know, I tend to make the really important words I think really jump out, like things you have to know, like ovary, uterus, fallopian tube, in capitals, and then I make some of the others in small letters, but you can do what you want. You can write them all lowercase, all uppercase, whatever you want to do is fine. You don't have to do exactly what I'm doing. When it's important that you do exactly what I'm doing, I'll let you know. So let's make an egg right here. Okay, let's see if there's anything else I need to say about that. We got the uterus, the endometrium. The egg is ready to start its journey. So once a month, actually, this ovary, it will be every other month, releases an egg. And it's kind of interesting to note here that there's empty space. The egg kind of floats out in nowhere land here. You know, it could escape. What if it got out and went around here? You know, then I guess it would never get fertilized. I suppose that happens once in a while. But generally, the little fingers like this, the fallopian tube has basically little fingers that pick it up, you know, go like this and grab it, pull it in. I know, not exactly, but so let's put it right here. Let's make this day 1. The picture we saw on day 1 where it's being fertilized, it's now a zygote, is occurring right here. So let's make the little dots around it for sperm. So a lot of times people imagine that fertilization happens here in the uterus, but in reality the sperm have to swim all the way up here through this tiny skinny tube all the way to the end. And of course the point in having that happen is to make it difficult for the sperm to get there, because you only want the strongest sperm to survive. So they make this long arduous journey, find the egg, and then one fertilizes it, and we have a zygote on day 1 right there. So maybe about day 2, maybe it's gone that far. It has to make its way down to the uterus. And then maybe day 3 here, something like that. Maybe day 4. Now, how is it moving? Well, you might think, well, gravity is taking it down. But gravity actually does not play a large role. The way they're moving is that there are little tiny hairs called cilia all along here. Now, of course, these are microscopic. This is way out of proportion, but we'll just make them large enough to see. So I'm going to circle some and point, write the word cilia, C-I-L-I-A. And cilia means tiny hair, or even I think it maybe means in Greek eyelash, little tiny hairs. Zoom in a little bit there. And so the cilia wave like this. They beat this direction, and the ones on top would beat down like this. So you have this kind of motion, beating down and swishing the egg down like this. Now what would happen if the cilia were not able to get the egg down? What would happen if it got stuck? This would not be a good thing, would it? Because this little embryo is going to go right on developing. So like day 3, we've got a few cells here. This is going to be the morula down here and the blastula. It's going to keep right on developing. And if it goes ahead and pops out of the zona pellucida and starts turning into a very tiny embryo, very soon it's going to get larger than the diameter of this tube. So you're going to get basically a baby developing right here for a short time. And that is not a good thing. It's extremely painful and in fact can be life-threatening for the mother. So any woman that suspects she's pregnant and is having sharp pains on her side, they immediately try to find out if she has what's called an ectopic pregnancy. And really the only thing they can do is just remove the embryo. I don't think they can successfully implant it down here. Okay, so by day 4, we're here. Day 5, maybe somewhere down in here. Day 6 would find it floating around, and for a short time it can float around in here. Now, if it doesn't manage to stick to the side and it floats too long, it's going to get flushed out, and there just won't be a pregnancy. But what should happen here, for the embryo's sake, is it should, by day 7, bump into the side and stick right there. And that's what the next drawing is going to be about. Now I've drawn it sticking right here. You know, maybe it might stick here or on the other side over here. It doesn't always stick right below where it comes out, but it may do that a fair percentage of the time, just kind of roll down here and stick. So the endometrium has been prepared by the woman's body. It's very rich in blood vessels, which are going to supply nutrients to the growing embryo. And we'll see exactly what happens here in drawing 26.